So hello everyone, my name is Yulin, and today I'm going to present my research, light-activated siRNA endosomal release or laser by Porphyrin Lipid Nanoparticles. So right now we know that lipid nanoparticle or LNP remains one of the best delivery platforms so far to deliver nucleic acids, as demonstrated by its clinical success of delivering siRNA, mRNA, as well as CRISPR Cas9 gene editing tools in human patients with good biosafety and efficacy. However, there is a fundamental challenge of the LNP system despite of its clinical success. That is, the majority of the RNA actually is being trapped in the endosome. So the poor RNA endosomal escape efficiency has hugely damaged the efficacy of the LNP. As you can see, for example, after LNP endocytosis, actually only 1 to 2% of the siRNA can be released into the cytosol. And previously, the researchers have also shown that by encapsulating siRNA gold conjugates into the LNP and image it with a transmission electron microscopy after cellular uptake, the majority of the gold siRNA conjugates were found in the endosomes, with a minimum amount being released into the cytosol. In another word, actually over 90% of the therapeutic potential of the RNAs is wasted because of this low RNA endosomal escape efficiency. Therefore, I want to develop porphyrin-based lipid nanoparticle to address this fundamental challenge. So first, we can see the structure of the porphyrin lipid at the bottom here. You can see that it's synthesized by conjugating the porphyrin molecule to the side chain of a lyso-PC. And you may wonder what is porphyrin. In short, we can consider porphyrin as color of life. The reason is because porphyrin naturally exists in the form of heme in our blood, as well as in the form of chlorophyll in the leaves. And it's a very natural biocompatible compound. And more importantly, it's a natural photosynthesizer. Therefore, if we introduce porphyrin lipid into the original LNP system, and it will enable LNP with the capacity to generate reactive oxygen species, or ROS, which can be used for disrupting endosomal membrane. And here comes the hypothesis of my research project. That is, once the LNP is endocytosized, because in those acidic organelles, the LNP will gradually destabilize and possibly dissociate. So some of the porphyrin lipid can partly translocate to the samely hydrophobic endosomal membranes. Therefore, in those cases, if we shine it with near-infrared light, then it can generate endosomal membrane-specific ROS, which can disrupt the membrane and allow the trapped RNA molecules to be released into the cytosol. And first, we prepared porphyrin LNP using a standard microfluidic mixing method, as shown here. And you can also notice that porphyrin LNP is synthesized by simply replace 10% DSPC from the original Onpattro formulation with 10% of the porphyrin lipid. And prepared porphyrin LNP displays a very homogeneous size distribution as shown by cryo-EM, as well as dynamic light scattering with a size around 60 nanometers. And it also maintains the optical properties from the porphyrin lipid. You can see there's a signature peak from both the absorption spectrum as well as the emission spectrum at around 670 nanometers. And also, porphyrin LNP is able to generate ROS upon exposure to light. So we can see that as we increase light dose, there is more ROS being generated. And more importantly, if the environmental pH decreases from 7.4 to 5.0, there is actually more ROS being generated, which is a useful property for our application because the LNPs, once they travel to the acidic compartment, that is the endosome, there will be more ROS that can be helpful to disrupt the membrane. And next, I performed a confocal microscopy to validate our key hypothesis, that is to visualize light-activated siRNA endosomal release. So as you can see here, the porphyrin LNP were incubated with cells for around 6 hours, and the siRNA was labeled with FAM and shown in green, whereas porphyrin lipid is shown in magenta color. So for the first row, we can see that before the laser irradiation, both the signal of FAM siRNA and porphyrin lipid is in punctate dots, suggesting they are trapped in the endosomal structures. However, once we apply 30-second light irradiation, we start to see a dramatic increase of the cytosolic siRNA signal, suggesting the successful release of the siRNA into the cellular space. And we have validated laser across broad cancer cell lines, including prostate cancer cells, breast cancer cells, pancreatic, lung cancer cell line, as well as ovarian cancer cell lines, indicating laser can be a general strategy that is widely applicable to many cancer cell lines. And more interestingly, if we zoomed into a single cell and image the cells with higher imaging resolution, we can actually find that porphyrin lipid is shown in the ring structures, as you can see here. And these ring structures enclose the FAM labeled siRNA very well. And similarly, if we shine it with light for 30 seconds, we start to see a significant increase of the cytosolic FAM signal, whereas the FAM signal from the punctate dots starts to decrease. So this data also is good evidence to show that actually part of the porphyrin lipid can translocate to the endosomal membrane. Therefore, once we shine it with light, the membrane-specific ROS can disrupt the membrane and allow the siRNA to be escaped. Moving forward, I did some in vitro luciferase knockdown experiments to validate whether laser can improve siRNA transfection potency as well. As you can see from the figure A, for the cells which were treated with Porphyrin-LNP plus light irradiation, it can achieve much better luciferase knockdown as compared to the cells which were treated with same formulation but received no light. And the quantification result is shown in figure B. Importantly, we can see that for the cells which were irradiated by light, there is no decrease in the cellular viability. So we know that this is a safe strategy to be used. And we also know that the decrease in the bioluminescence is not from the decrease of the cell viability. And next, by establishing dose response curve, we can see that Porphyrin-LNP plus the laser strategy can significantly reduce IC50 of the siRNA to around 4-fold less as a result of improved siRNA endosomal escape. Moving forward, we also performed a proof of concept in vivo experiment. So in this experiment, instead of encapsulating siRNA in the LNP, we encapsulated gold nanoparticle into the Porphyrin-LNP to visualize the gold nanoparticle later through the TEM. And you can see, so we have subcutaneous tumor-bearing mice, and the Porphyrin-LNP GMP were intratumorally delivered to the mouse tumor. And 6 hours afterwards, the tumors were irradiated by a 71 nanometer light at 50 joule per centimeter squared light dose. And then the mouse were sacrificed either immediately or 18 hours later, and the tumors were taken out and processed to be imaged under the TEM. And as you can see from the TEM image here, so for the tumors which receives no light, the majority of the gold nanoparticle actually are found in the endosomal structures. So it's similar to what we see with previous confocal results that suggesting they are trapped in the endosomes. However, as soon as tumors were irradiated by the light, we start to see the decrease of the membrane integrity. And there is a clear trend that the gold nanoparticle is escaping in the locations where the membranes are destabilized. And at a longer time post-irradiation, we can even find some gold nanoparticles randomly distributed in the cytosolic space, which are not enclosed by any membrane structures, suggesting the successful and more robust release of the gold nanoparticle. And using image J, we also quantified the portion of gold nanoparticle in the cytosol versus in the organelles. And the result here showed that as long as we apply the light irradiation to the tumor, there is around 3 to 4-fold enhancement of the gold nanoparticle endosomal escape efficiency, supporting that laser strategy can also work in vivo. So to summarize, this project developed a Porphyrin-LNP that is based upon a clinically used Onpattro formulation by replacing DSPC with a porphyrin lipid. And secondly, porphyrin lipid can translocate to the endocytic organelle membranes during the LNP endocytosis. Thereby, it can generate endosomal membrane-specific ROS upon the near-infrared light irradiation. And also, a laser approach significantly improves siRNA efficacy by facilitating the endosomal membrane disruption and allowing siRNA endosomal escape. With that, I'd like to thank all my lab members and our key collaborators, as well as generous support of the funding agencies. And also, this paper has been recently published. So if you are interested, feel free to check out more details in the following link. Yeah, thank you. I'm happy to take any questions.
I have a question, Yulin. So can you quantify the siRNA endosomal escape efficiency?
Yes, actually, I have another slide. Actually, if we want, it's doable. So what we did is that we captured the confocal imaging across the Z-stack and reconstructed into 3D structures. And with that, by processing it using a software called Imaris, it will be able to capture the cellular structures as well as the organelle structures. And by doing that, we can specifically create a mask that only show the cytosolic portion of the siRNA. And by comparing the siRNA intensity towards a standard curve, as shown below, that is also generated by creating a 3D confocal imaging dataset using the FAM-labeled siRNA alone, then we will be able to tell the concentration of the siRNA before and after laser irradiation. And then you will be able to calculate the siRNA endosomal escape efficiency.
That's cool. Thank you so much.
Divya, I have a question, if there's no other questions. It's a similar question, Yulin, and thank you very much for a very interesting presentation. Some very exciting results coming out of this study. How do you quantify the gold nanoparticles subcellular distribution using the TEM imaging that you described in your slides?
Yeah, that's a great question. So here you can see that actually we captured a lot of transmission electron microscopy images. And as you can see in the top left figure here, so each white square represents a zoomed in TEM image. So by zooming in, we can see a lot of gold that is either distributed in the cytosolic space or it's in the endosomal structures. And by passing those images to the image J and set intensity threshold, then you will be able to select specifically the gold nanoparticle that is in the cytosol or the gold nanoparticle that is in the endosomal structures. And you can sum up all these numbers across different views, then you will be able to get the endosomal escape efficiency per one cell. And then I did that calculation for many, many cells. That's how we get that number.
Excellent, thank you.
Here's my second question. Does light have any influence on the LNPs without the porphyrin lipids?
The answer is no. That's also a very nice question. And so, as you can see from this slide, we also prepared the LNP that does not contain porphyrin lipid. That is original Onpattro formulation, as shown in the top figure here.
Mm-hmm.
So you can see that even though the laser was applied, there is no significant change of the cytosolic FAM signal or the FAM signal in the organelles. And we also did another control that is, we have the LNP that only contains porphyrin lipid, but it does not encapsulate FAM-labeled siRNA. And you can also see that even though we applied the laser, we didn't see any green signal coming up. So we know that the green signal is not resulted from the ROS generated by porphyrin. So it's not a side product. It will be the FAM-labeled siRNA.
Thank you so much.
I have maybe one more question. Given the high potential and the success of your studies to date, maybe we could just get a sense from you of what the limitations are of this technology and what other studies are needed, or what this suggests for future research initiatives that are needed as well.
Yes, thank you, Diana. That's a wonderful question. So the limitation, first of all, it's a technique that is based on using external laser, and light has some intrinsic limitation, that is penetration depth. So you can imagine right now we cannot apply laser to treat some deep-seated tumor. But in our study, we should also mention, notice that because this strategy, we are not trying to use light to create a lot of rods to kill the cells. We just want the rods to disrupt the membrane. In other words, the light dose required would be significantly lower than the typical photodynamic therapy applications. So that part will kind of elevate the light, the penetration depth limitation. And also, right now, it's a preliminary work to demonstrate how those external stimuli trigger work in the LNP. So you can imagine in the future, if we can apply some, for example, ultrasound sensitive lipid or X-ray sensitive lipid, then those lipids, if they can also translocate to the endosomal membrane, then you would imagine if you apply the external stimuli, which does not have penetration depth limitation, that can also work to improve the RNA endosomal release.
Okay, great. Thanks so much.